Hey everyone, I'm in a scientific laboratory at CSIR National Institute of Oceanography, and it's very much confidential to mention the lab which I am really present. Well, I wanted to show you how a typical laboratory in an oceanographic research institute look like, and the videos is mostly related to science, so people who are really into science or shows any kind of interest in science can listen to this video. Before going into any further details, I wanted to show you how the different part of a oceanographic research institute's lab look like, because it's not a single lab that function in oceanography. There is multiple lab which combine together to work on a specific functions. So let's get into the different parts. So right now I'm in the molecular biology lab of this research institute, and this lab is really a different kind of typical molecular biology lab what we witness in several highly advanced laboratory system in the world. Because first of all, this is the oceanographic research institute. So in biological oceanography, when the scientists study biology, so they prefer to study the environment and the animals of the ocean, and based on that they provide different kind of reports. So in some of the cases they require the molecular biology, but most of the cases they do not. Many of the cases they study biodiversity by counting simple the number of the species which are present in the ocean with respect to different parameters of the environment, like temperature, like pressure, like the salt in the ocean, different acid or base present in the ocean, the presence of viruses, bacteria and other animals. So this is how the oceanographic research happens. And what they use, how they use the molecular biology. So you can see this is a machine called RT-PCR, the real-time PCR, as you have heard during the COVID timings. So this machine is very useful for quantifying the DNA present in the samples, most of the animals or bacteria or viruses. So this machine, if you can design the primer against the DNA, this machine can amplify, make a multiple copy of that DNA and show whether there is a presence of certain amount of DNA sequence in the sample or not. So let me clarify in a simpler way. For example, let's say you have 10 different of fruits in a shop, like 10 difference of mangoes, and mangoes in generally they are same, but when it comes to the DNA sequence, the mango will be different, because although they are same mangoes, they are variable in terms of taste, variable in terms of colors and textures. So when you study the DNA sequence, if you have a certain set of primers which can make a copy of the DNA, so when you make the copy of different DNA of from the mangoes, so there is a constant region in the DNA which are very conserved. I mean they are throughout the life, throughout the evolution, they are constant, and this constant region will have certain sequence same in all the cases, but when it goes to different species, there will be a variable region, like where the sequence are different. So in the different sequence, this machine will give you DNA copy of the DNA sequence, and you can go for DNA sequencing. So, and this machine you see is a DNA sequencing machine. So these machines are called ion torrent semiconductor sequencer, and so basically what that does is they make DNA into small fragments, and those small fragments are getting again smaller, and as they grew smaller, so there is another process happen in that machine called polymerase chain reaction, based on which you get a certain sequence, and the machine will count that sequence, whatever the small sequence they are making up, the machine will count it and give us which one is variable and which one is constant. And this machine is a general PCR. So what it does is it also make the copy of multiple DNA sequences, but comparison to this machine give you the accurate values, how much your DNA is present in the sample, how much they are amplifying. Here the scenario is different. Here only you can set the temperature at different cycles, and this will give you the ideas. This machine is called a gel dock. So once you are done with PCR or any kind of DNA or RNA amplification, so we have to run a gel. So the gel is called agarose gel electrophoresis. So you see this, this is a gel, and this gel is run within this gel. These are called gel casting wells, and this is the machine. If you switch on, it gives electricity, and the DNA are negatively charged. They move from the negative side to the positive side based on electricity and size differences. This is how they work. And one more thing, so this machine, which is not looking that great, but these are called the servers. So they are very much essential when it comes to high processing system of DNA sequencing. And we always made some sticky notes about different protocols that we use for our experiments. So we basically do homogenization. It means you lyse the tissues, and then you lyse means you crush the tissue samples from the ocean and then grind them to get the DNA through several procedures. And we have gel, these are called gel ladders. So ladders means you get a DNA, so which size they are falling in, and we have experimental ideas. Okay, let's get into this machine. This machine is called centrifuge machine. So what it does is, so you put some let's say some sample and chemical together in a small tubes. They're called centrifuge tubes, and you put into it, so they rotate. So they rotate in a circular way in a very high speed. And so what it does is, so it will concentrate every samples to the bottom of the tube. That is called centrifugation, and it separate different component of the sample, like big component and small components are separated. This particular thing is called laminar airflow. So when we work on some bacteria, we wanted to get away from the surrounding, because in this surrounding there are a lot of contaminations present, like a lot of aerial DNA or bacteria, or like small dust which can destroy your reaction, because in dust there are several many particles which is very much essential. They can interact with your enzymes, proteins and chemicals to completely change your reaction scenario. So we have like there are like light and flow system, as you can see. So light and flow open, and then we wipe out with ethanol or methanol, like we have ethanol, and then we use that. Okay, so this thing is called another kind of laminar. So we have light and UV. So UV is actually dangerous. You see how it looks like. And UV is dangerous to skin because it can causes cancer. So be careful whenever there is a UV light presence, because UV light, what it does, it can change your DNA sequence, and once the DNA sequence is changed, it cannot be revert back. It cannot change to the original, and a fault protein may produce from the DNA, which can causes problems to your body, your normal functions. And this machine, again, I wanted to say, this is called a UV spectrophotometer. So spectrophotometer uses light. So light can pass through certain chemicals and give a signal. So for example, you have a clean water and you pass a light, so light will pass through it transparently. But when it comes to a chemical, let's say you put sugar into it, or you put some dye like blue dye, so this machine will give you different signals, because blue is high in color and red is high in color. So let's say some chemical produces a certain kind of light like blue, green or red, so they give different kind of signal based that is recorded by this machine. Okay, our famous and most importantly we all know, so this is called a microscope. This is a fluorescence microscope. So what it does is, so there is this thing called lamp. So this lamp provide light to this machine, I mean the eyepiece of the this microscope, and this microscope has samples kept in here. We have different zooms, so we see the image through this area. And there are like camera options, eyepiece, and then eye and camera, and only camera. You can visualize this on the PC. So that is how it works. Okay, now let me go to a different part of the lab. What we have exactly, we have a cooling centrifuge here. So this machine gives you temperature, very low temperature, like 4 degrees centigrade or less. And this is a shaker. So this machine will shake and mix different kind of chemicals. This is called hot air oven. You can actually put things to dry with high heat. This is also temperature controlled incubators. Incubators give you specific temperature to store samples. This is another incubators where we have 37 degrees centigrade. We store different bacterial samples. This thing is a -20 refrigerator. -20 is a standard refrigerator in different scientific institution because it can store various kind of chemicals and your samples for a very long time, like 1 year, 2 years. We even have -80 refrigerator like this, so that is for many, you can store things for decades. Okay, this is a micro oven, so we generally prepare some samples like medias. We can put into this, and once we switch on, like start it, so it will boil and give the things. Weighing balance. Weighing balance is one of the most important things in science, because any preparation of chemicals give us a standard sort of measurement to it, and this thing is called molecular water system, ultra pure water system. So what it does is, so it removes all the different ions and chemicals from water and gives you the pure water without any other chemical contaminations. Okay, this is an ice machine. So we have water supply, and this water may turn into ice in this machine. So once it switch on, it gives ice in this area. You can collect and use it for your work. These are the ice boxes. This is called a water bath. It gives you temperature control water bath for incubation of sample. Well, incubation of samples mean you keep the sample in certain temperature condition for a very long time. That is called incubation. Incubation helps in achieving the desired function of any particular reaction. Reactions means experiments. So that is a water bath, and it is a dry bath here. So dry bath means also there are like different blocks. We have very small tubes to put into the blocks, and this machine gives you certain temperature which can be used to achieve a desired incubation period. This is a pipette. So we have different pipette in microliters. Microliter are very small amount of measurements, because we don't need like 1 liter or 2 liter or 1 ml, 2 ml. We need very small amount of chemicals to study in laboratory, because in laboratory you study everything at micro scale. And this is a pipette holder, and we generally have tissue paper for wiping out things. Okay, next we go to so this is a self-frost refrigerator. In generally we have 4 degrees centigrade or -20 degrees centigrade refrigerators, and we have chemical storage system where we store our chemicals. So for example, this is our, according to the alphabets, we have stored our different chemicals. And just hold on, I'll show you different part of our lab. Okay. So this is my sitting place. In the sitting place we have our tables. We can decorate it in our way. And okay, my keys. What I'm going to show you is a lab called autoclave. We named it as autoclave lab or wet lab. Okay, so in this lab, first thing I'm going to show you is, this is an incubator for different phytoplankton culture. These are the small plants from the ocean, and these plants are stored in some kind of media, and this light they use to photosynthesize and grow together. Okay, there are so many species we have collected, and this thing is called a plankton mesh. Planktons are very small microscopic animals, and they are collected using nets only. You cannot collect it using bigger things. We have different fridge for different purpose. Okay, This thing is called an autoclave. So an autoclave is, what it does is it produces steam to purify your sample, to give decontamination to your samples. This is a wet lab look like in Oceanographic Research Institute, because we have motor to filter the seawater we collect during the sample. Then we prepare metal granule columns to purify nutrients, and then we have acid to acid dispenser, I mean, for using the acids. So in autoclave lab, generally we filter the samples, we do oceanographic storage of samples, and then some cultures. Okay, so this is how a autoclave lab look like. Okay, we have advanced laboratory systems called proteomics. This big machine you see is called proteomics system, and there are like machines inside. They are called flow cytometry. So there are like liquid, when they flow through it, it gives certain kind of signal to quantify different chemicals or bacteria or cells present in that sample. So this is all about our laboratory, which is present here. And with this, wrapping up this video. Okay, I forgot something else. So we have another instrument that is called confocal microscope, but I cannot show you that because the lab is generally very much locked from outside. I'll just show you how the lab looks like. So this is called, you see this? So nothing is visible. So we have confocal microscope for the work in the laboratory. So today's videos is about this much, like an introductory video. Maybe hopefully in next upcoming videos I'll show you how we do research, I mean what is really research and science in the everyday life we do here. Like some of the experiment I will record and I will show it to you, because it's fun to share your stories to common people when you are isolated from rest of the society. The society doesn't know what is it really looks like to do science. So it's better to communicate with the humans that are present beyond science. To, I mean, everybody is a scientist. I mean, other people also have scientific temperament, although they are not present in the laboratory. Like a farmer knows when the rains will come and when he will put seed into the field. So many stories are out there, and I will gradually cover up everything, whatever I known so far. I will also share my journey, how I came here to work in this lab, and how the government of India is paying everything for my expenditure. Yeah, blah blah blah. And thank you so much for the video.